today's talk is mostly focused on assessing the genetic stability of iPSC-derived allogeneic cell therapy.
So Parimal, welcome to this session, and we look forward to hearing you present your scientific data and then also the discussion later on the collaboration between India and the US. So over to you.
Thanks, Narendra, and thanks to Susan and Gaurav for your introduction. So hi, my name is Parimal Pande. I am originally from Nashik. I completed most of my education in Nashik and got my B Pharm from University of Pune. And after completing that, I came to US to pursue a master's degree in biotechnology from University of Houston, Clear Lake. After that, I worked for a CRO, which was specialized in providing genomic services. It was back in Houston, and I was there for 3 years, where I got to learn all the lab techniques for starting from Sanger sequencing to all the next generation sequencing. Working for a CRO, as you know, you work on a project and you just finish with it. But as a scientist, it actually bothered me a lot. So I decided to pursue a job in research. So I was looking for opportunities in academia and in industry as well. So I got a job at Boehringer Ingelheim Pharmaceuticals in Connecticut. I was working in the toxicology department there. I was leading a genomic lab. And while working full time, I also completed industry-sponsored PhD at University of Connecticut. So my project was basically developing in vitro model for steatosis and looking at epigenetic regulation of cytochrome P450 gene. For the last few years at BI, when I was working, I supported AAV-based therapeutics mostly. So they have the gene therapy portfolio. So I supported that in terms of safety assessment, genomic stability. There were a few lentivirus-based projects assessing the instruction site analysis and genomic stability. And based on that, I started working at Johnson & Johnson last year. So I'm at the Springhouse campus, which is very close to Philadelphia. Here at J&J, I'm in the genetic toxicology department, supporting both cell therapy and gene therapy. So for the gene therapy projects, I support, again, insertion of AAV. For the cell therapy, I support genomic stability of allogeneic cell therapy, where you use CRISPR. So today's talk is mostly focused on assessing the genetic stability of iPSC-derived allogeneic cell therapy. So I will be talking about assessing genetic stability of iPSC-derived allogeneic cell therapy. All right, so cell therapy is usually iPSC or primary cell derived. So you all are aware of autologous cell therapy, where you have a patient, you collect your cells from that same patient, perform genetic modification, and give it back to the same patient. So this is the common practice. Genetic editing can be performed used commonly with a lentivirus-based vector, or it could also be performed using a CRISPR-based method. But this is like you have a patient and you produce one drug. So it's time consuming, it's expensive, and usually not preferred because it's more personalized therapy. But sometimes if you have a healthy donor, you can perform modification, you can knock out a TCR or perform some other editing methods to incorporate CAR. With that, you can produce a drug that can be given to multiple patients. But in that situation, the starting material is primary cells. But in the iPSC-derived cell therapy, your starting material is again going to be primary cell. It could be immune cells, such as like a T cell, gamma delta T cell, or fibroblast. Those cells are again converted to inducible pluripotent stem cell using several Yamanaka factors. And at iPSC stage, you perform editing. And these edited cells are expanded, banked, and then becomes a drug substance and drug product, which can be given to multiple patients. So in this situation, this therapy is off the shelf, so it's readily available. It can be stored at a various location and given to the patients right away, where in the autologous cell therapy, it is uh, produced and it takes time. So there are some advantages with allogenic cell therapy compared with the autologous cell therapy. So if you compare allogenic versus autologous, there are several advantages versus disadvantages. In the autologous cell therapy, it's patient specific, so there is a lesser chance of rejection by the immune system. Again, there is no risk of graft versus host disease, and repeated dosing is possible if the therapy fails because of some reason. But there are many disadvantages such as the high manufacturing cost, starting material variability. Uh, we're getting background noise. Can you mute your microphone? Um, Again, the product is heterogeneous. So in terms of assessing stability, it's a big challenge. Time taken to manufacture these product is very long. And these patients undergo several lines of therapy before they are given these autologous CAR T therapies. So these immune cells of these patients are exhausted. So several times they just fail to expand, and there are just not enough cells uh, to be given to the patient. And again, cytokine release syndromes and neurotoxicity have observed in some patients, which is again a bit challenging. So CRS can be manageable, but neurotoxicity could be difficult to manage sometimes. So to overcome these challenges, allogenic cell therapy is being developed, for which large-scale manufacturing is possible. Reproducible manufacturing batches are homogeneous. They can be stored for a long time, and you can perform multiple freeze thaws. Donors can be screened upfront for any genomic stability-related liabilities. And again, you can perform self-editing in these cells so that they can evade yeah, immune-mediated rejection. Again, some of the disadvantages are like limitations, such as the in vivo expansion is limited and repeated dosing is just not possible with these therapies. And since you are performing editing using CRISPR-based method or any other method such as like TALEN, zinc finger, or transposons, in that situation, insertion, mutagenesis, translocation, and genomic stability-related risks are higher. So as I said before, you start off with a donor material. You screen donors to make sure that they don't have any existing liabilities such as like translocation or any aneuploidies. And also they are not carrying any communicable diseases. So if there is an insertion of any virus, then it can get activated over the period and it could cause a genetic liability. So these healthy donor cells are modified using Yamanaka factors such as OCT4, SOX2, KLF, and c-Myc to make them into inducible pluripotent stem cell. This is a one-time programming event where you convert healthy donor cells into inducible pluripotent stem cells. And this stem cell bank can be used for multiple projects, multiple targets, and they can be stored for a long term. These iPSCs can be modified with your CAR of interest for your target of interest. And again, this editing process is one time for that particular target. Once the editing is done, this is the most critical stage where you need to perform genomic stability assessment, because if you are performing single knockout knock-in process, then the chances of translocation and genetic risks are low. But in some situation, you might want to knock out multiple targets, such as knock out T cell receptor, knock out MHC1, MHC2, knock in multiple CARs, dual CARs. In that situation, chances of on-target translocations are very high. So assessments of genomic stability have to be done at this stage. So these edited cells are clonally selected. So a single clone is selected and that clone is expanded to make CD34 positive cells, and then they become whatever cell types you are interested in, such as like alpha beta T cells, NK cells, or gamma delta T cells. So assessing stability at master cell bank of post-edited iPSCs, as well as the final drug product is very important in this situation. So
Parimal, can I ask a quick question there if you go back? Sorry. When you're banking the cells, you already you have not differentiated the iPSCs when you're banking the cells. So the same bank could be used to differentiate and making them into different kinds of cells. So whether the same bank can be used to make NK cells versus T cells or anything else for that matter.
Absolutely, yes. So there are two types of cell banks. So these are unedited iPSCs and these are edited iPSCs. So the edited banks can be differentiated into NK or gamma delta or T cells.
Gamma delta or T cells. Okay, got it. Thanks.
All right. So let's look at the consequences of gene editing. So whenever, is there a question? All right. So if you're using a CRISPR-Cas-based method, so you have a Cas9 and a guide RNA, which binds to the target strand where it makes a cut. So there are two requirements. The first requirement is identification of PAM sequence, which is a protospacer adjacent motif. So Cas9 protein has an ability to scan genomic DNA for the PAM sequences. So whenever it encounters PAM sequence, it looks for complementarity with this guide RNA. So once these two things are identified, the Cas9 makes double-strand cuts at the target strand and at the non-target strand. So the double-strand break is repaired with two pathways. The one pathway is a non-homologous end joining, which involves incorporation of random nucleotides in this double-strand break. And the other one is a homology-directed repair, where you provide homology template, which could be either double-stranded DNA or a plasmid. So you have to have a homology with the homology arm, where based on the homology-directed repair pathway, this gene of interest is incorporated into this double-strand break. In the non-homologous end joining a pathway, which is very quick and occurs very fast, and it occurs multiple times. So whenever there is a PAM and there is some sort of homology with the guide RNA sequence, the Cas9 starts making cut up until this guide RNA complementarity is lost, or up until this PAM sequences are completely destroyed. So usually it creates 50 to 100 base pair of insertion deletions, which we usually want, which causes some knockdown of that gene of interest, where a homology-directed repair occurs. So on-target repairs are something we want, which includes substitution, insertion, deletion, and also on-target integration is something we want. But there could occur chances that this homology template is incorrectly incorporated, or if this guide RNA binds to some off-target sites, so there will be off-target integration of your gene of interest. So instead of having only one copy, there will be multiple copies of your gene of interest. There could be structural variations such as large insertion, large deletion, translocation, inversion, and duplication. So these are the liabilities that we need to be addressing whenever we perform our double-strand breaks, or whenever we are using this CRISPR-Cas-based method. FDA has put forward a draft guidance last year for genomic editing in order to assess the consequences such as like clonal expansion, increased proliferation, and these cells gain genetic fitness if we perform editing, and if there is an off-target edit that leads to, let's say, increased proliferation or increased survival of these cells. Insertion mutagenesis, and that could in future lead to clonal expansion and oncogenicity. So in order to assess these consequences, FDA suggested to assess some attributes such as on-target editing, efficiency and frequency, off-target editing, again, frequency and locations, genomic stability assessment for small and large structural variation, copy number assessment for the gene of interest, which we are incorporating, as well as the whole genome copy number, because off-target editing could lead to gain of copy number or loss of copy number, and the copy number assessment needs to be performed. And they suggested several assays such as unbiased and biased method, and certain parameters such as using multiple method. Those methods should be sensitive, and use of appropriate controls so that those methods are accurately performed at CROs at your labs. And again, using multiple donors. So the suggested assessments for off-target edits are discovery methods. So use of multiple methods. So if you're using a biased method, that is your first initial screen. So if you're designing a guide RNA to knock out a specific gene, then you use a different in silico method to perform to identify whether that guide RNA is going to bind at different locations or not. So that is your first way to identify good guide RNAs which have lower liability. But again, this in silico method is a biased method. So suggestion is to use unbiased method. And in order to perform unbiased assessment, the suggested methods are either biochemical or intracellular. In the biochemical assessment, the suggested methods are digenome sequencing, change-seq. And for the intracellular assessment, the suggested methods are guide-seq and induce-seq. So in the discovery mode, you perform different methods such as in silico assessment, biochemical and intracellular assessment, and perform a Venn diagram analysis where to find out a combination of method which off-target sites you are identifying. And based on that, you rank the method using bioinformatics approach if the identified off-target sites are of high risk, medium risk, or low risk. So if these off-target sites are in the intron or somewhere in the genome and are not affecting any gene, then they are of low risk. But if they are in the transcription start site, or if they are in the exons of genes that are known to cause oncogenesis and that they are mentioned in the databases such as the Cosmic database. So in that situation, you need to perform validation because there could be a false positive as well. So you design primers against the predicted off-target site and confirm them using various methods such as like RAM-seq, ITR-seq, duplex sequencing, or TLA, targeted locus amplification. And with that, you perform your risk assessment to show that whether that off-target site is going to potentially cause any off-target effect. So I will briefly go over these methods. I won't touch upon all of them because there are hundreds of methods to perform these analysis. But some of the commonly used methods, such as like guide-seq and digenome sequencing, I'm going to mention here. Guide-seq is an intracellular method. So you transfect the cells with a ribonucleoprotein complex, which is your Cas9 and guide RNA complex together, also known as RNP, together with a double-stranded oligodeoxynucleotide. So the sequence for this ODN is known. So whenever there is a double-strand break, this ODN gets incorporated into that double-strand break, and you can design primers to amplify this sequence and find out what is the neighboring junction. So with that, you will find out where the double-strand breaks are occurring, which is a very good method in predicting off-target site compared with the digenome sequencing, where you collect genomic DNA from the donor cells, incubate them with a ribonucleoprotein complex for like 8 hours. And after that, you perform a whole genome sequencing and identify these off-target sites. In the digenome sequencing, it's an in vitro method. There is no homology-directed repair, or sorry, the non-homologous enjoining repair mechanism. So you get a lot of off-target sites. And again, if you compare with a naked DNA versus a DNA that is wrapped around a histone octamer, you're going to find out a lot of non-specific off-target. But digenome sequencing is to cast a wider net to find out more off-target site instead of going with only few where you could miss some off-target sites. So these are two very nice methods that are commonly used. In terms of validation, again, you can perform amplicon sequencing, so design amplicon against the predicted off-target sites. But amplicon sequencing produces a lot of background. And in order to avoid that background, these two methods are developed, known as like RAM-seq and duplex sequencing. So RAM-seq uses a RNA base and a blocking moiety. So this RNA base need to be cleaved off using RNase H2, which gives it a lot of specificity. And it upon amplification, you basically have very low level of background noise through Ramsey. Again, with the duplex sequencing, it uses duplex tags. And during amplification, if there is a mutation that is occurring or a random base is incorporated, then that random base is removed during data analysis. So this is very specific. In terms of translocation assessment, some of the traditional methods such as like G-banding are commonly used, but G-banding has a cutoff for like 50 megabases. So small structural variations occurring that are lower than 50 megabases are less likely to be found out using G-banding. And it's very likely that we will see such structural variations in this technique. Some newly designed techniques such as like DGH assay, this is provided by a chromatid Inc. This is one of the companies. And spectral karyotyping, these are good. But again, these are not great methods to assess structural variations. Whole genome sequencing is again a benchmarking standard which you could use because you can detect base pair changes. But again, data analysis cost, that is expensive. And at what stage of analysis you are applying this whole genome sequencing is critical. Again, whenever you're looking at a whole genome and looking at a lot of mutations, it's going to be a bigger challenge in terms of data analysis. And again, it opens up a lot of questions you cannot answer. So if FDA asks you a question, or if a regulatory authority asks you a question like, okay, how can you explain these mutations, then it's really a challenge. Some other newly developed methods such as like optical genome mapping are quite interesting, and I like those as well. So in this method, you isolate a genomic DNA and you use a dye which incorporates into a 6-mer repeat that occurs consistently into the genome. So based on that, you perform analysis on this Cepher machine, because these patterns are consistent across the genome, and bioinformatically, you can build a genome. So if there is a structural variation such as translocation, inversion, duplication, those repeat sequences are switched. So with a computational methods, you can assess that. And this is very quick, like within 24, 48 hours, you can perform a data analysis and get your results. But one of the biggest disadvantage is the throughput. You can run only 6 samples at a time, so which is still a big challenge. So if you look at the whole end-to-end manufacturing process, starting with the donor to inducible pluripotent stem cell, editing, then intermediate cells, and the final cell type, there are various stages where you could go wrong, not just in terms of genomic stability, but there are many ways things could go wrong. So there are several assays that need to be performed at each stage in order to make sure that there are no genetic liability that you are carrying forward in this stage. You are culturing these cells ex vivo. So over the period, you are accumulating several mutations, and which is pretty common. So there will be several common carrier mutation, as well as there are mutations specific to the donor. So you need to perform a genomic stability assessment at a donor stage, at iPSCs, at post-editing, and at the final cell bank, because you can screen out mutation that are specific for donor that occurred at the iPSC stage and that occurred because of editing. Because with that, you will be able to answer that, okay, these are, this is how we got these mutations. And you can perform a further risk assessment, whether they are of high risk or low risk, either a paper-based argument or performing additional experiments. So in conclusion, these iPSC-derived cell therapy products, they undergo extensive culturing, and they can undergo several freeze thaws and stored for a long term. So they tend to acquire a lot of mutation. You need to make best judgment and perform assessments at each stage to perform genomic stability assessment and perform your argument based on paper or additional experiments. If you are performing multiple editing, then the chances of translocation and genomic instability are high. But if you are performing extensive characterization, then you can move this product forward, saying that we perform, this is actually an assumption that we are producing a drug which carries less liability. But again, whenever you put these products into patients, there are patient-specific reaction, and you could see clonal expansion in some patients, so which is always going to be a risk. So again, you need to use a weight of evidence-based approach, use multiple methods for off-target analysis, because only one single assay at this moment is not going to be sufficient to predict the off-target effects. So yeah, thank you.